Thanks, Josh. That was great. I'm going to try to use this clicker just in case. Yeah, so I'm really excited to talk to everybody. As Josh said, I am interested in what I'm a chauvinist about proteins as well. I think are the most important and fun to study biomolecules called proteins. So I just have a brief introduction. We know that proteins do really important things like protect us from diseases, allow us to breathe. This is a cool one. It allows the butt of an insect to glow, and so this is luciferase, which is found in fireflies. So as humans, we're not often just okay with all of these amazing things that biology has given us, these amazing protein functions that biology has given us, and we will often want to modify or change the activities or the abilities of proteins to do things to suit our needs. But this is a really difficult challenge. And so one way that we can try to address this challenge is by using computers. And so what I'm highlighting here are a few examples of some changes to proteins that computers have made. All of these were done using this software suite called Rosetta, which was developed by David Baker at the University of Washington, who is my postdoc advisor. And so one of the things that Rosetta has been used for in the early days was to predict the structure of proteins from just the sequence of proteins. This is something that many of you will have heard about recently because this is what AlphaFold also does. But this work was started not just by Rosetta, even before that, but Rosetta was very successful at that for a number of years. This is a pretty staggering thing that Rosetta was used to do, which is to make a protein fold that doesn't exist in nature. So you say, I want to make a protein that looks like this, and you feed that into the software and it gives you a sequence, and lo and behold, it folds into this particular structure. Another thing that Rosetta has been used for is to make completely new enzymes from scratch. Of course, we would really like enzymes to do myriad things for us, and Rosetta has been used to do this. And I felt like this was a pretty relevant example, considering the flu is obviously now sort of rearing its ugly head again. We just had a COVID pandemic. The software was used to design a completely new protein that could bind to and inactivate the flu. So this just goes up to 2010. In the last 15 years, computational protein design has been used to do many other things. But I just wanted to highlight what I thought were some of the really amazing things that the software can be used to do. Now, I'm not shown on this slide anywhere. So what do we do? And so what I also focus on protein design, but as I say here, I try to simplify things by actually making them a little bit more difficult. So what we're looking at here are amino acids that are not found in nature, but these all have, and so this is 63 out of over 200 amino acids of this type, which have been added to the genomes of organisms from E. coli all the way up to mammals like mice. And so any of these amino acids that you see here, which have functional groups, chemical functional groups that are not found in the naturally occurring amino acids, can be placed into proteins. And so when I started my postdoc, I wanted to ask the question, can these unnatural or non-canonical amino acids be used to design interesting proteins? And so, I'm going to be in the first author place in most of these papers, in all of these papers, with David Baker at the end. So all of this stuff was done using Rosetta. And it turns out that Rosetta, even though it wasn't trained on amino acids not found in nature, can totally design proteins that contain them and design proteins that actually do important things. And the only thing I really want to highlight in this slide is that we have computationally designed proteins in gray in these two images here, and solved crystal structures in blue and in salmon here. And this is showing the accuracy that Rosetta was able to design proteins that contain amino acids that aren't found in nature. So one brief slide on what we do here. So I've continued to work on this sort of thing at ASU. We've changed the amino acids that we focus on. The two amino acids that I'm showing here are both fluorescent or fluorogenic, which means that in this case, if you bind a metal ion to this amino acid, you will get an increase in fluorescence. And here's showing some rational design studies that we've done Again, Rosetta is able to design proteins with unnatural amino acids really, really well. This is a crystal structure solved in our lab here. And we were able to rationally engineer proteins that respond to metals and increase fluorescence in both of these cases. So, okay, all of that's really cool. That's all I'm going to talk about the research that we do here. Ask me more about it if you would like later. But so I wanted to end by talking about the 2024 Nobel Prize in Chemistry, which was awarded to, among others, David Baker, who's my postdoc advisor, for computational protein design. And then, as I mentioned, AlphaFold, the people responsible for that are Demis Hassabis and John Jumper. They were the other two people that were awarded the Nobel Prize this year. And so back in December, a few of us well, 200 of us from my postdoc lab went to Stockholm for this celebration. And so I'm showing here the outside of the concert hall in Stockholm, in downtown Stockholm, where the Nobel Prizes are given. This is the only photo that I have from inside the concert hall. And of course, neither of these people are me. This is my dear friend, Neil King. This is right before the awards were given. David got something like 15 people that could actually be in the room with him. And clearly I was not on that list, but my good friend Neil was. So this is the inside of this building right before, David was sitting in one of those chairs waiting to get his prize. And then I do have a few pictures to prove that I was actually there. So here's the former boss in the Grand Hotel. I'm at the end. Again, my dear friend Neil King, grad students and former postdocs with David, including me. This is a photo I took in this beautiful auditorium in Stockholm right after the Nobel lectures with David, Demis, and John. And then this is the closest that I'll ever get to a Nobel Prize, after being in Stockholm. This is at the Nobel Prize Museum. Any of you can go take a picture next to a ginormous Nobel Prize medal. And so with that, I'm happy to answer any questions here, but please also reach out to me at this email address if you want to discuss anything that you've heard about today more. So thanks for the opportunity.